Hello everyone. Today I am going to present in brief the main findings from a recent paper entitled Chemosensory Neurons Modulate the Response to Oomycete Recognition in C. elegans, which was published in the journal Cell Reports. Oomycetes are a group of fungi-like eukaryotic pathogens which infect both plants and animals. The most popular and well-studied of this group is Phytophthora infestans, which causes the late blight of potato that led to the Great Irish Famine in the 19th century. While several animal oomycete infections have been reported, there is only little information about host immune responses as well as oomycete-derived signals that are recognized by animal hosts. However, we now have a tractable experimental system which can allow us to understand animal oomycete infections better. In 2018, our group identified the oomycete Mesocetiopsis humicola as a natural pathogen of C. elegans, which is a well-established model for studying host-pathogen interactions and innate immunity. We reported that the nematode responds to the oomycete by inducing a specific set of genes known as chill genes or chitinase-like genes as shown by a gradient of GFP in the hypodermis of the worm, and these are required to antagonize the infection. Interestingly, we also observed that even autoclaved pathogen could induce the same response, suggesting that chill gene induction could be an outcome of pathogen recognition rather than actual infection. Intrigued by this observation, we decided to just wash our culture plates with water and collected the liquid following centrifugation and filtration. When the extract thus obtained was added to worms carrying the appropriate transgene, they started glowing again, confirming that chill gene induction was indeed happening in response to pathogen recognition rather than infection. The extract thus provided an opportunity to uncouple pathogen recognition from tissue damage that happens in infection. We performed transcriptome analysis of extract-treated animals and compared it with the transcriptome of infected animals. A significant overlap was obtained between the two, as pathogen recognition is also going to be the first part of the actual infection process. Based on this overlapping dataset, we describe in this study the oomycete recognition response with chill gene induction as its hallmark. This early detection of the pathogen by worms is definitely advantageous, as extract-treated worms showed reduced pathogen attachment and enhanced survival upon infection as compared to non-treated controls. The main body hypodermis of C. elegans is a multinucleate syncytium, so we were intrigued about anterior localized expression of CHIL27 within a single cell. We argued that this localization may reflect signal transmission from the anterior side of the animal, which is dominated by neurons and glial cells that could readily participate in pathogen detection. Also, a subset of OR genes that were not expressed in hypodermis were previously known to be exclusively expressed in neurons or neuronal support cells. We therefore decided to test the possibility that neuronal signaling may regulate detection of M. humicola by C. elegans. Towards this, we chose to use TAX2 mutants which exhibit compromised chemosensory function. We found that loss of function of TAX2 strongly reduced the induction of CHIL27 upon extract treatment, thereby highlighting a potential role for sensory neurons in mediating the response. Inability to detect the pathogen and launch an immune response also made these mutants hypersusceptible to oomycete infection as indicated by increased pathogen attachment and reduced survival in an infection assay. To conclude, our study provides a clear evidence for the existence of oomycete recognition in C. elegans and highlights the requirement of inter-tissue communication, especially between neurons and epidermis for animal defense to oomycete infection. Thank you.